The human brain is arguably one of the most complicated objects in the known universe. It has billions of neurons and trillions of synaptic connections, and we are collecting more data per unit time on this organ than in any other that I'm familiar with. Being able to analyze that data and process that data and be able to turn it into new knowledge is a real challenge. And it's a challenge that we can't do unless we bring high-performance computing to bear on dealing with that big data challenge.
Historically, research infrastructure has looked like wet labs with benches, spaces like that for people who need, kind of someplace to go mix chemicals. It's been these very capital-intensive, large-scale projects that are very specific to particular disciplines. Increasingly, across all disciplines, computing has become a fundamental tool, and so the need for a robust, centralized infrastructure that's available to all those researchers has become paramount.
So realizing that the University of Virginia has an enormous neuroscience community that are all heavily dependent upon data science techniques to be able to process and analyze and model their data, we recognized a major opportunity here to augment current research computing infrastructure with dedicated hardware for the use of the brain sciences community. And we have constructed this thing called the NEO system, which is specifically developed to accommodate high-performance computing for brain-related data.
So the NEO system is a compute cluster. It's 24 standard Intel x86 64-bit servers. This is an actual node, a cluster compute node. So this is an entire compute server, 2 CPUs, 384 gigs of RAM in here. It also has a GPU server, which has some very beefy, has 8 NVIDIA GPUs. These are just very nice high-end video cards, essentially. They don't actually have video card outputs because they're used for very specific kind of deep learning processing. At the moment, we have 16 petabytes of storage that we're attaching to this that can be used across the platform or deployed across the platform. Within that, we're hosting a couple of different environments. The first that we're going to be providing is traditional, what we call HPC, which is high performance computing. On that same kind of platform, people run what's called high throughput computing, which again is just a very large number of jobs that can be run through. They don't necessarily have the parallel aspect. But then in addition to that traditional environment, we are providing, in the process of building out what we call a orchestrated containerized environment. What they're super handy for is they're a wonderful way to move computational environments around transparently. This is a way for researchers from multiple institutions to develop a shared environment to do their research.
There are a number of different examples where a platform like this is going to be really critical. We have several of them going on here at UVA.
My lab is interested in studying the brain structure, particularly the circuitries that are formed between neurons and and other neurons. So we have 2 different electron microscopy approaches that we're currently using. One is the transmission electron microscope, which generates data, basically images. Images in very detailed, high resolution, as I said, this 2 nanometer resolution images, which can cover about 50 micron by 50 micron areas. So each image is actually quite large. And we collect a lot of these images, and my lab generates thousands of electron microscopy images per year. So storing the images and archiving them is actually one issue. The second approach, the Connectomics approach, uses a different data set, which is basically stacks of electron microscopy images collected on a scanning electron microscope. Several hundreds and thousands of these individual images make up a stack, and then we go over them and identify and trace and color individual dendrites, terminals, and axons over subsequent images to make our three-dimensional models of the brain structures. So you can imagine that that requires a lot of space and processing power. Being able to manipulate those image stacks real time is a real challenge.
The brain data that we collect with human neuroimaging involves a lot of different steps to be processed from the time it comes off of an MRI scanner, for example, and is something that then you can go and take and analyze and publish your results with. I hope that this becomes a go-to system for all of our neuroscientists here at UVA, as well as being a major educational resource for our young students here who are interested in not only the brain sciences, but also data science.